So it's sags, but
yeah. Okay, let me know when you're clear and
I'm clear.
All right, hydraulics coming down.
Ooh. It's not how I thought dinner relief was gonna go.
Awesome, man. I don't even think you broke a spine.
Yeah, that is very well done indeed.
Okay. Yeah, you can bring me back up.
So yeah, we have about 40 minutes approximately left on the bottom here, give or take a little bit, and then we will have a post-dive call just to give our shore-based scientists a heads up at 6:15 Hawaii Standard Time.
Get out in front of you there.
Fish.
Ah.
A little jelly.
Yeah.
Try to get that guy rolling.
Okay, copy that. He looks very small.
I can't hear you, Jim.
Do we want to get the ship moving east again?
That'd be okay with me. Yeah. I don't know if we're going to get him rolling.
Yeah, he's pretty far out there.
Yeah.
So yeah, that looked like a jellyfish, Andrea.
Sorry, video.
Co-pilot, does it look like this ridge bears due east?
I can't really tell until...
So, Daniel, did we put any hypodermic needles on the ship in the lab?
Yeah.
I think so, yes. So we want to inject them?
So when that urchin comes up, can you inject it with the alcohol in addition to putting it in alcohol, but inject it right through the mouth?
Will do.
Do ship move, please. 50 meters, bearing 090 degrees, decimal 2 knots.
And
Perfect, thank you.
And Nav, I'm not sure. I can't see on HiPAC. Did they drop a target?
So yeah, this is
for that sample.
Maybe not. Should I do that now?
Yeah, we should do one before we get far. I even came northeast a little bit.
Should I say anything specific?
Yeah, there's a naming convention.
so this is going to be now. This is going to be Spec 03 Bio, and I have the metadata here too, as if you need it.
You tell me if I need it. I haven't done this before.
Wait, there's a in the Nav notebook or the binder. There should be a naming convention that we use typically for the targets.
It's the same. It's... Yeah. There's a sheet in there that explains it.
Okay.
And these groups of the Novadynia Sea Star, this is something that we've seen here on top of this
the biggest thing, and then we can
small ridge and a few places where you have close to the edges of the ridge. But yeah, not much for corals. We have seen a few of these small chrysogorgids
Kind of ran off there.
and anemones, but other than that, this debris of unconsolidated rubble doesn't appear to be a good habitat for corals or sponges.
Anything out there, Roland?
Nope. Nothing we haven't seen.
Gotta bring out your upper swing arms.
Copy that.
So yeah, most of what we see here on top of it is, well, lower densities of mammals. We have seen a few clusters of these aggregations of the sea stars, as well as of sea pens and isolated colonies of these chrysogorgid, those yellowish colonies of corals. And then several rat-tail fish have also cruised the area.
It's a sea star.
Yep.
You want to go check it out?
Yeah, let's.
We can probably reach that.
Any chance we can take another look at that sea star if we're close enough?
Yeah, we're heading that way, Chris.
We'll let him go, Roland.
Yeah.
How's that light pool rolling?
Further out would be better than what I'm seeing currently.
Yeah, I don't think I can.
The
don't think I can get
front
much
pool
help
is
from
very
it.
hot.
Okay, rolling. I can square it up once you get in. That's going to be hard to image.
Does that help?
Yes, that did help on this particular image.
Josh, can you maybe tilt me
Turn
up
the
just
lower
a
ones
touch?
off.
Gotcha.
Got both arms.
Full zoom.
So your upper surrounds are deployed. I just turned them off though.
Copy that.
Yeah, I'm still not sure.
Where's Chris Ma?
Yeah, he might be. So yeah, Chris has already provided a genus ID here as Terraster. And yeah, if the other Chris Ma hasn't gone to bed yet, I'm sure he'll chime in with an ID.
Should lasers cross it
Depth
as
right
we
here
pull out?
is
Copy.
And video's clear.
686 meters.
Lasers are on.
So yeah,
if you
this
can.
animal is a
Yeah,
little less
go
than
ahead.
10 centimeters
Just gonna hold
across.
it while we're here. Thanks. There you
Okay,
go. Okay.
full out.
Next video.
Give your camera back.
Thank you. You have caught up.
So I have.
Interesting little white ledge you're coming up to.
Yeah. Have you seen that C pen, I believe?
We have.
Okay, both of those.
Oh, that clear one in your lasers we have not. Up here.
That's what I was. There's a fish down there too.
Yep.
I'm gonna go ahead and partial for both of them.
Oh, it's a coral.
Ah. Let the fish go.
Start
Just
at the
hold there,
base.
rolling. I'll tilt up when I land.
Yeah.
There you go.
Land.
So Dan has stepped
Step
out
down to the
to
base.
grab a
Gotcha.
quick bite to eat for dinner. So I'm going to try and play the best biologist that I can, just because it's fun. And I'm going to try to identify that as an Iridogorgia.
Yeah,
I'd like
go ahead
to,
and tilt all the way to the top.
Consider this my quiz.
Oh, sorry.
Hi, Jonathan. This is Andrea. You got the right family, which is great.
Woohoo!
More like a Chrysler. Yeah.
Ah,
Got some streamers off the top.
I'm trying to learn.
You can also see a brittle star on the left,
Oh yeah.
and then there's the shrimp inside. Looks like there's possibly a shrimp or a squat lobster in the middle of that colony.
Video, we're gonna do a quick pilot change.
Copy that. And we're going to do a
You
video
can see
change.
it's one cloth up to the left. It's probably a shrimp in the genus Baffipalmanella, I believe.
Thank you, Andrea. So we're just going through a quick personnel change on our ROV team. So we'll get moving here in a little bit.
Hey Dan, this is Chris Maw.
Oh, Chris Moss. So Dan has actually stepped out to get a quick bite to eat for dinner, but I'd love to hear your assessment of that last sea star we saw.
Okay. This is interesting. So it's a pterasterid for sure. I think it's Hymenaster, but I'm not sure which one it is, or because I don't think it's any of the ones we've seen already. I think Mbari actually collected something similar to this a few years ago. It has a very different texture and a different color, and it's white as opposed to the other ones, which are purple. So, but it's also the texture is a little unusual relative to the other Hymenaster species. So, yeah, I mean the entire genus Hymenaster is one big sort of complicated situation. So, it's someone's PhD project to be sure. But
let's do another identical.
But unfortunately, so it's Hymenaster. That's the best. It's Hymenaster, or possibly even one of the other more obscure pterasterids. Like, there's a one called Hymenasterides that I've never seen a lot. But it has a red spot on the oral surface, and so I wouldn't be able to tell you without looking at the underside. So, so are you actually just in the control center, or is this?
Yeah, so this is Jonathan. I'm in the control center. Dan has just stepped out to get a quick bite to eat, so I'm trying to play my best biologist that I can, and consider this my biology quiz for the day.
Okay, yeah. So, but yeah, so it's so I guess the long and short of it is it's a slime star.
Interesting.
And as with many of the things that we're seeing in the Hawaiian area, it is poorly understood, and I would think that it's part of a larger, more complicated issue. But my guess is that if it was collected and brought up in a big collecting box, it would be filled with mucus by the time it got to the surface.
Okay.
On that nubbin.
That's basically what those do. They produce mucus as a defense. Well, hence the name slime star.
Oh, okay.
So the moment they get put into close quarters or they're stressed out at all, they will just go crazy. And I've gotten the collecting box full at the top of a collecting event, and it's just filled with mucus. So hence the name slime star.
Great, thanks, Chris Ma.
Anyway, have a good one. Good night.
You too. Good night.
Before you leave, I got to ask you. So this thing had a skirt, if you will. Oh, did he get off?
Yeah, Chris. So. I think we lost him.
Yeah.
Let's zoom in.
Sure thing.
The light's not great.
Stand by.
I think Bobby's working on it.
That's some beautiful imagery right there. Wow.
If we could center him up.
And not just of the shrimp, but the...
Take your mini suits too.
Okay.
You know, it looks like this might be a heterocarpus, but I'm not a shrimp expert. I don't know if there's anybody out there who might know.
Yeah, Bruce, this is Heterocarpus levigatus.
Yeah, there was a question earlier about was there trawling up at these seamounts and all that. I don't know if shrimp fishery was at this particular location for a short while. Actually, it's continuing a little bit. There was an intensive development, deep water shrimp
kind of nice to get a lot of contrast
this species and one other. And a lot of research went into it. Some of the knowledge about the fishes, deep water fishes of the Hawaiian Islands comes from trawling for the shrimps that was conducted by Paul Strusaker, who then looked at the fishes for his dissertation. It turns out that the populations of these shrimp are easily knocked down by a fishery, and the fishery really wasn't sustainable to any degree. So it stopped. It was mostly conducted with traps, though, not trawling. And the fishery went on for a short while, and then the resource kind of diminished, and it became economically unfeasible. Every now and again, somebody comes back and does trapping in the appropriate areas for the shrimp. They do it for a year or so and then stop. So it's an interesting fishery history for that type of shrimp.
All right, thanks, Bruce.
Off to the right. Go check out another sea pen.
Snappy little arm.
Would you like to take a look at this?
Sure.
I think we've seen it before. Just try and keep moving.
Yep. I'm just going to keep going up slope, and you let me know if you see anything.
Yeah, I'll do my best. Thanks. But it definitely appears that as we keep on going shallower and shallower, we're starting to see a little bit more of the lithified carbonate, which is kind of interesting. I don't know if that's a function of the more unconsolidated coral skeletons being removed due to current flow, or if this is a different regime of paleo reef coral growth, or I'm not sure what we're seeing right here.
Do we want the fish? I think it might be the same thing we've seen.
Yeah. Get stopped, and then we can take a quick zoom.
Sure.
Okay.
So that was another macrid or rat tail or grenadiers as a common name. Seems to me from a non-expert observation that most of the animals that we've been seeing in this more shallow portion of the dive can be kind of described into like a handful of species. What is that? Is that two animals or just one?
Looks like two.
We were wondering the same thing, if it's attached by a filament or not.
And what exactly is this that we're seeing?
Looks like a siphonophore.
Oh, okay.
All right, it's pretty far away now.
You're heading downhill.
Yep.
Siphonophore the broken half.
Okay, now you got my toe in there.
Coming up on the end of the move. Should I just keep it
come back over, Bobby?
Should I keep us moving, or you guys want
I
to?
think you can keep it moving.
Yeah.
It seems to. I think we need to go a little bit north.
A little bit north, okay.
For the next one.
Like 10 degrees north or more?
15, I think, should be good.
075. Bridge nav.
Yeah, keep coming.
Yeah, let's do a move now. 50 meters, 50 meters, bearing 075, decimal 2 knots.
Okay, I'll come over, Bobby.
Perfect, thank you.
Yeah, so if anybody is joining us towards the end of this nice long extended dive, we're currently at a depth of 673 meters. We actually reached the bottom at about 1,280 meters. So close to about 600 meters of total relief that we've gained throughout this, and over the course of that ascent, we traversed through the oxygen minimum zone. We've seen a neat distribution of these vertical separations of different kinds of biological communities, as well as some very interesting geology. We were able to collect one geological sample at the very beginning of our dive that will be critical in determining the absolute age of this seamount, as well as 2 different biological specimens. So we've gone from a mainly heavily manganese-encrusted, probably basaltic volcanic edifice into a lesser manganese-encrusted volcanic edifice, where we were able to make out some primary flow structures, such as some nice pillow lavas, and even image this beautiful elongated pillow lava tumulus that extended down the flank of the volcano. Currently, we've reached the summit ridge of this seamount that rises about 4,000 meters off of the seafloor, and the landscape has dramatically changed from a mostly manganese-encrusted volcanics into this kind of a graveyard of coral debris with sparse, what seem to be lithified carbonate rocks. Again, biological community has changed in its composition as we've gone and reached the top of the seamount.
Sorry, I checked out for a little bit to grab some dinner, and I'm just updating here. It sounds like you guys saw a few new animals there while I was gone.
What's that thing? Just a shadow. Looks like there might be something in that hole there. Check it out. Okay, snap zoom video.
And that's gonna be a
An enemy.
yeah.
Okay, let's come wide.
Yeah, it's this debris field is quite extensive. Yeah, we started seeing it at a depth around 750 meters or so, and yeah, extending here throughout the entire top portion here of the seamounts apparently.
So yeah, Dan, as you were gone, we kind of transitioned in the landscape to being predominantly softer, unconsolidated debris into this what seems to be more lithified fossil reef type of look to it. So again, not sure what to make of it, but it looks like to me that this at one point in its history was at a shallow enough depth that could have formed an extensive carbonate complex here. Just never got a chance to grow into a flat-topped atoll.
Getting pretty far out.
Might be another slime star there.
Should we take a look at it, or?
No, just
No?
if you're,
Okay.
if you need something for a video.
We have time.
Yeah, certainly do.
Yeah, very few corals here at the top. These chrysogorgid corals are the, we just see far and few in between. And even on this substrate that's a little bit more consolidated. No, that's an urchin. And yeah, we did get a good image of that before.
Okay, zoom out.
So, Daniel, this could be something that's called micropyga. You can see those really long tubes coming out of the top of it with little knobs on the end.
Yeah.
And this is about the right depth for that. It's very similar to echinotherids, but it could also be an echinotherid. I'm not sure. And if Chris is watching, maybe he'll weigh in.
Yeah, thank you, Chris.
Welcome in to move. I'll give it another one.
Yeah, I think the next one can probably be 060.
Okay.
A little shrimp. Oh, he's gone.
Bridge nav. Move 50 meters, bearing 0.
I'll bet you guys feel a little bit like that sea pen we just passed.
Decimal 2 knots.
No, not at all.
Perfect.
Yeah, I'm more like
That was the most tired sea pen I've ever seen in my life.
we are more like that sea star to the right with her arms up in the air and pumped.
We didn't get a chance to ask you how you fared out there with the bad weather the past couple days.
Yeah, it was. There were some rough moments, but most of it, yeah, just we're eager to get in the water and dive. But it was a really interesting exercise of taking a look at the weather forecast, and just basically as far as we could ever travel, it was the same bad rough seas around us, so there was nowhere to hide. And yeah, that's one of the common problems up here, is that the land features here. We have a couple of these very tiny atolls. The Pern Hermes Atoll is about 100 miles north of us, but yeah, that provides zero protection if you have a ship even going close to shore. They're very, very small islands. There's no lee, and this is all open ocean, even close to the islands.
Sorry, Tara?
There was a fish to the left.
Yeah. That one?
No.
Yeah. Coming left.
Over there. Just
It was interesting. We got a whole bunch of comments from folks who had chimed in, had logged in to see video, hoping to see the dives, and all they had was the video of the camera from the side of the ship, and people were saying that they were getting seasick from watching that camera, and some people developed fascinations for watching that. It was
zoom in.
I had some very interesting email exchanges about people sharing that. Oh, that's a phenomenal. Oh, that's a beautiful image.
Oh, sorry.
Bruce, if you still hear us, don't leave yet.
Okay, just as I started to get up, I saw the fish, so I'm here. Don't know what this is. It's obviously very different from this. Oh, what is this? Is this a Coelurynchus with the shovel-like snout? Is this a Coelurynchus eratrum or one of the other ones?
Yeah.
It could be a Eratrum, or what about Dorisis?
Yeah, that's the other one I was
zoom
trying
out.
to think
I
of.
can try
Yes,
to
this
get
is
closer.
definitely one of those, and I can't
didn't wanna
remember
scare
what
'em
the
off
characters are. We
first
can
time.
look in the Hurl guide. But yeah, we've got our first Coelurynchus, one of the more shallow-living Macrura genera.
Do you need me to move on, co-pilot, or?
You got time.
Okay.
I've changed my heading to come around and look at you.
Okay. Oops. Zoom in.
Can you guys put on the lasers, please, after you're finished
Go for it, Bobby.
with the great images here?
Yeah, copy that, Andrea. Yeah, well,
That's full.
Yeah, this is excellent. Yeah, this genus has a flattened, almost sturgeon-like snout and has a little different, probably feeding mode than some of the others, more oriented toward the bottom. And in fauna, maybe Andrea knows more about that than I, but this is excellent.
Tara?
So Bruce, the Eratrum has got the sort of white patch on the dorsal fin, if I'm not mistaken, and Dorisis doesn't, at least from the images in the
Yeah,
guide.
we'll let him drift
So
off.
just on that basis,
Okay.
I'd be more inclined to call it Eratrum.
Okay, that works for me. I'm not keeping up in terms of looking up the references, so sounds good. That was excellent.
So
Sorry, where do you need me to go?
I'm looking closer towards our path, so.
Okay. Oh boy.
yeah, there's a question here, Tina. Yeah,
Come up and go
none of
lateral.
we have not seen any live scleractinian corals up top here on this debris field.
In the heading.
060.
Okay.
So you don't need to worry about my heading. You just get into my view. And then you'll be set.
Yeah.
I actually think that is the first volcanic outcrop of rock that I've seen in the past 100 meters or so.
Yeah. And
Gives you an idea of the approximate thickness of some of these carbonate layers, so not very thick at all.
yes, and these dense aggregations. So yeah, what Jonathan was mentioning is that the color changes. So the dark, that's volcanic or basalt, and the lighter colored substrate here, that's calcium carbonate. That's fossilized reef.
Just making up a thin veneer on top of this seamount.
Yeah.
So Jonathan, are we looking at lithified sediment, or are we looking at actual reef remnants here? And if it was sediment, I mean, where would it even come from, since it's right on the top?
Yeah, that's a good question, Chris. I don't think it's sediment. I think it's actually lithified reef. So anytime that we've been zooming into animals, you can kind of catch a glimpse at some of the calcium carbonate as well, and it kind of looks like it's been, in place or in situ and lithified. So there are different kinds of structures that remind me of, you know, any of the other drowned reef complexes that you see, or
Next one's gonna have to
any
cut
of the
more.
exposed reef complexes. So yeah, we're zooming in right now, and you can kind of see some
heading
porous material, maybe some remnant structures as well. But I don't think I can make out any kind of sediment.
No, yeah, we're just zooming in on a Liponema sponge here, very distinctive for their short, pointy tentacles.
30? Or 45. We can do 45. Yeah, 45 is fine.
Yeah, and I agree. Just from this view, it does look like fossilized reef.
Yeah.
Let's do a move, please. 30 meters, bearing 045 degrees, decimal 2 knots.
Okay, zoom out.
Perfect, thank you.
Forward.
Oops, sea star. I think.
So actually, we've gotten shallow enough. We can go till about 610 local.
Copy that, Nav. Thank you.
Okay, let's zoom in.
It looks like another Terraster asteroid.
Okay, zoom out. Thank you.
So yeah, our navigator has just informed us that we'll have until 10 minutes past 6 here on the bottom, so just have about 13 minutes of bottom time remaining.
I gotta push ahead, Tara.
Okay, no worries.
Looks like a pyrosome, or?
Yeah,
Yeah, pyrosome.
Pyrosome.
Kind of interesting. For the past hour and a half, we've kind of been, going over this sediment, seeing the same fauna, the sea pens, a lot of Novodinia, occasionally Echinus.
Yeah, again, this whole concept of pioneer communities where something gets established here and then it kind of spreads out from there. Novodinia
some more.
reproduce, of course, but when you're on the top of a seamount like this, you're not going to be sending a whole lot of larvae up the water column. They're just going to wash right off. And so they've got to have a way in which they can settle fairly close.
Look, everybody, it's volcanic rock again. Yay!
Yeah, yeah, it looks like we're getting here very, very close to the shallowest portion of the seamount. We're just at 655 meters. And yeah, another change in the substrate, going from calcium carbonate over to basalt.
The path looks to me like we want to go east.
And yeah, this is a remarkable abundance here of these Persingent sea stars.
So you okay going east, or still want to go a little more north than that?
Yeah, I don't have a great feel.
Yeah, that's a beautiful view here of this dense, very dense aggregation of Persingent sea stars.
Sounds great.
Bridge nav. I'd like to go 30 meters, bearing 080, decimal 2 knots.
You want to zoom in on that guy?
Yeah.
Thank you.
We have a comb jelly there, or Tina 4.
Might actually be a tiny bit too close. I think it's too close to focus on.
Yeah, okay, let's go full wide.
Yeah, this is just a very remarkable image here of this very dense aggregation. Maybe we can take a look at that one yellow coral on top.
Okay. Let's zoom in partial. I'll try to keep it framed up, but
Oh, on the top?
Yeah, that's fuzzy stuff. Yeah, there's two
Go ahead.
corals here, yellow on the left and then on the top
Where's
if you
it?
could zoom in. Oh, something different on the right here as well.
Yeah, we'll get you both, Andrea.
Oh, I'm losing it. Pretty far away. Sorry, Tara, hold on.
No problem.
Just don't want to run into any of these.
Yeah, so definitely a plexaurid, but and there's another chrysogorgid right next to it.
I'm just going to set my toe down. Tilt up. Okay, zoom in.
So one
It looks like it's Canthagorgia, I think, but not sure the one on to the right after finish with this one.
Yeah, we'll finish this one, and then we have this Ascaris schemata. It's ophiuroid on top, and then we'll take a look at the coral to the right there.
The pink one.
Okay.
Yeah, just in front of us.
Tilt down, I'll get there.
Looking down 90 degrees.
Okay, we don't have a whole lot of time.
Yeah.
Okay, got what you need, watch lead.
Yep.
Thanks.
Excellent. So a few corals here. And yeah, very nice dense aggregation here, but most of it of these sea stars.
Another one of those fish. We saw it
The,
earlier.
the Coelurynchus, that's another one. That's Tokiensis. That's a fish on the lower right.
Yeah, we'll get
Sighting of that.
you a view here.
Get a nice one. He's pretty well lined up for you.
Okay, zoom
Well,
in.
I just can't get out of here. I've been trying to see all these interesting fishes. One interesting thing about this fish is it hasn't been recorded from the Hawaiian Islands except from the Hurl records before. And we saw something very similar at Hurl dives down in the Line Islands. But there was something in a paper by Tomio Iwamoto and I think Nigel Merritt on New Caledonian
We'll come out partial and get the rest of the
Macquarids that made a comment that this actually may be a species complex, a very similar species, but there aren't enough specimens to really work it out. So there's definitely an interesting taxonomic problem with this type of fish.
I think we should hold off.
Just hold.
Yep. Looking back at you.
Bridge nav,
Okay, I gotta
this
come
move
up.
we're going to hold for the time being. Yeah, that'd be great. We'll plan to leave the bottom at 10 past the hour.
And see which direction.
Yeah, this is quite remarkable.
Okay,
This
I'll touch it.
higher density of sea stars kind of throughout the top here.
More drift. We're awfully shallow.
Okay.
There's nothing on sonar.
Is this the top of it?
Yep.
Well,
Yeah, so
Turn right. Top of your local.
I see.
10 or 15 more meters to the top.
Yeah,
So close.
yeah, looks like we're going to be reaching the very, very summit of the seamount here.
We can certainly try.
Unfortunately, we have 5 minutes left, I believe. Nav, if you want to put a ship move in, you can, but we're not going to make very far.
And yeah, there's a little bit of discussion on this on the chat room, and a few people commenting on these dense aggregations of sea stars. And apparently, Dr. Scott France had seen something similar on the
Bridge nav, let's do a
in the northeastern
move
Atlantic
30
and some of the
meters, bearing
canyons there.
090, please. Decimal 2 knots. Perfect.
There's a very interesting discoloration to these volcanic rocks. I'm not quite sure what it is yet. Can't tell if it's just altered material or some sort of bioprecipitate, but
Well, that's a pretty sight.
You guys found the Sea Star Garden.
By the way, what to do on the way up, or to get started up, just tell the bridge.
Once we're off the bottom and comfortable, we'll bring the ship half a knot forward over ground.
So I don't need to do anything initially with the ship.
No, except bring them.
Yeah, this is quite remarkable, these dense aggregations of here at the top.
yeah, perching themselves on a
Yeah, sure.
seemingly local little high on this kind of boulder up and in the current flow, I guess.
Yeah. Yeah, but Chris was kind of discussing there. It's very interesting that here towards the top we kind of see all the things that we think are favorable for corals and sponges, but we only see a few, but we see a lot of these sea stars. So, yeah, a lot of times they might be recruitment limited, that even if you have the perfect conditions, they got to get there in the first place. And so until you have enough corals to get there in a critical mass to kind of, you might have the perfect conditions, but
You're sort of on your last object anyway before we have to set up.
Take a look at this guy.
yeah, this is a very, very pretty scene to end with.
Zoom in.
Oh, good.
Oh, he's pretty neat.
Oh.
Let's
This crab is named Sirdomaya smithi. He's a really fascinating crab. And all those spiny arms. He's great looking.
Yeah, good spot again.
Nav, let's end this move here, please.
Thank you, Jim.
And if we have last second there, it says one coral to our right.
Okay, we're just gonna get a zoom in on this crab. Let's go in video.
Okay, now you. That's good.
Thank you.
Hold here for a moment.
Yeah, and you can see some barnacles there on some of his legs.
Oh, cool.
Those are yeah.
Don't they have an ointment for that?
Yeah.
Probably a real turnoff for the lady crabs.
I like the two horns.
That's for the young viewers out there. That's what happens if you don't shower.
Or molt.
Hopefully we don't run out of water on the ship.
Might be trying to molt.
All right, what was the coral, Dan?
Right, to the right.
Pull out here, I think.
This will be the last thing we get a shot
As we
of.
pull out here, it's interesting to note these Brisingid asteroids all clustered
Right
on
there.
this little
a raised part of the bottom, and they were that way before, and it's just another comparison to the ledge getting up off the bottom to the higher flow.
Yeah, excellent point there, Scott. And so yeah, we are about to run out of time here.
Start bringing your.
So we get one last view here.
Zoom video.
Another plexaurid. But yeah, as we're about to run out of time here, this is
very well, but
absolutely a remarkable last scene. Yeah, thanks, Dan. Yeah, remarkable last scene here. Very beautiful. The top here made it to the.
Yeah, I need to set up your swing arm still. And
okay, I'm gonna get a little closer.
Our setup's gonna be pretty.
This is our final depth here is 649 meters.
And a perfect setup for me to do swing arms.
Okay, let's try it again.
Yeah, and it's been a remarkable dive. Really appreciate everyone for staying on. Our shore-based scientists. It's been really remarkable. And then, of course, thanks again to our pilots and videographers. Done a spectacular job today. And, of course, the whole crew of the ship agreeing to let us dive late today. But I think we got a lot extra out by just adding these couple extra hours of dive time. And we covered, an enormous depth gradient here. Close to 650 meters in elevation gain. And yeah, it's going to be some very important data coming out there on what might be determining some of these communities and shaping them. We definitely saw some differences in the dominant species and different depths. But interestingly, we did not see this void of life throughout this oxygen minimum zone.
Time to tilt the camera up and get a quick shot at the tip.
I think we do.
We're cheating.
Great.
We're cheating.
Thank you.
Make it up.
And yeah, beautiful ass.
Do you have any idea what this coral is?
All right, Dan.
Okay. Let's zoom out.
Okay.
Who are you asking, Chris?
Yeah,
Turn your
do you
head
have any
left.
idea what this coral is?
Line up with the ship.
Oh, I like that it's paramurciid. It doesn't really look like an acanthogorgiid to me, at least from the characters that I'm seeing. The fact that there's an
You're going to go right down
astroschematic
that way. I believe
ophiuroid on
it's
it
going to be a
lends support to
left
that.
turn for you. Maybe a right
I've
turn.
seen lots of paramurcia that have yellow
is right,
pigment in the tissue with purple sclerite.
Why don't you start going starboard
Seeing
and
a
we'll see.
tinge of purple there along with yellow, so
Yep.
all of that suggests some kind of
So I'm gonna come up
paramurcia
some.
or close paramurcia relative to me.
Okay, anyone got an eraser? I'll erase my chat room comment.
Do you want me to come up with you at this point?
There's a permanent record, Chris.
Okay, sorry, go ahead.
So you're going to make your heading opposite me.
Okay.
And then just push out.
Okay.
Yeah, we're
So yeah.
above you nicely, so push up.
So as we're just leaving the bottom here, I just want to once again thank everyone, our particular shore-based scientists, as well as everyone here on the ship. It's been a remarkable dive. We'll take a quick 5-minute break and then maybe do our post-dive call maybe at 6:20.